Okay, grade 10s, we are now going to go through the 3-page worksheet on plant tissues and organs. It's a new one that I made this year just because you are at home and you probably need a little bit more guidance on all these tissues and organs and how they all fit together. All right, let's have a look and I'll explain some things along the way. Okay, so the first part I think you can go, it's pretty easy, all right? So any type of plant tissue whose cells contain chloroplasts. You remember that word that combines parenchyma and collenchyma. If they contain chloroplasts, so it's chlorenchyma. Dividing tissues in various regions of the plant, meristematic. Thin-walled cells, which generally have a storage function, like storing sugars and starch and water, parenchyma. Plant tissues whose cells are thickened, it looks like these cells are thickened with extracellulose at the corners, collenchyma. The term for various tissues filling up the inside, the interior plants, ground tissue. Irregular shaped cells whose cell walls are thickened with lignin and are found in pear tissue and the shells of nuts, sclerids. Those little irregular shaped cells, and it looks like they've got little tunnels going through the cell wall. Long tapered cells whose cell walls are thickened with lignin and form part of the water transport tissues. So that would be tracheate, xylem tracheate. Because these sclerids here, okay, they are sclerenchyma, and then you also get the long sclerenchyma, on the one side of the vein, for example, but it looks the same as xylem tracheate, but is what they do that is different. Xylem tracheates transport water. Sclerenchyma fibers would have the same shape. I haven't asked that here though. But they have the same shape, but they are for strengthening. Right, number 9, living sucrose conducting cells without nuclei. Although I think I probably shouldn't have said conducting. I should maybe have said cells that help the conducting cells, so sieve tube cells. I will make a note of that. Cells which assist in the functioning of the sugar conducting tissues, companions. Hang on. Okay, is correct. 1.9. Read carefully, Mrs. F. Living sucrose conducting cells. Yes, no nuclei. Sieve tube cells. I think I was thinking it said with nuclei. Then the cells alongside them that help with the functioning of these sugar conducting tissues, inflow, the companion cells. Okay. Right, then I gave you 2 micrographs, photographs of plant tissues, parts of plant tissues that have been stained, different plant organs, and they were both used and observed using the light microscope. Which transverse section is the dicot root? This one is the root. All right, so it's section Y, dicot root, section Y. How do we know? Because the xylem is in the center and it's in the shape of a star, like a 4-armed, whatever shape, number of arms star. And the dicot stem is X because they are separate veins, separate vascular bundles of veins in a ring around the edge of the stem. All right? You can stop and start this video as you go along and check yours. Identify the structure, ring G, so that's the vascular bundle, the vein or vascular bundle, cross-section through it. 2.3, tissues A to F in micrograph X. So we are looking at epidermis. Yes, I know it's not very visible, but we know this now. Collenchyma, sclerenchyma, phloem, xylem, parenchyma. Okay, you can go and stop that. Explain how you would be able to differentiate between tissues B and C in micrograph X. B and C. B is collenchyma, C is sclerenchyma. Collenchyma, cells in transverse section would look like their cell walls have extra thickening at the corners. Sclerenchyma, they would be evenly thickened walls in transverse section. Okay, and they would stand up red because of the lignin. All right. Anyway, I've got brackets. It's just to remind me what B is, but it means that I don't actually give a mark for that. It's just so that we've got in our heads, okay, what is this? All right, it just makes marking easier as well. 2.5, state the respective functions of tissues A, D, E in organ X. So A is epidermis, D is phloem, and E is xylem. So no mark for what they're called. Epidermis protects the stem from the environment from too much transpiration, as it has a waxy cuticle, etc. The phloem to transport food, sugars, sucrose through the plant. This should say xylem. Let's put that in. The xylem is to transport water and mineral salts from the roots to the rest of the plant. So only a mark for the function. Identify tissue F and write down the number of the label from micrograph Y, sure, we'll read this carefully, which points to the same tissue. So F is parenchyma, and 1 in diagram Y is also parenchyma. This is probably some kind of sugar or something that's stored in those cells, or it could even be starch granules. Okay, it's been stained up. All right. Tissue 1. Explain 2 ways in which tissue A from micrograph X would differ in plant or organ Y. So tissue A is the epidermis. There is no epidermis in this because this is part of the section through the root. So you've got to think. So you've got to compare. So, sorry about moving up and down like this. So A is the epidermis, and in the stem has a cuticle and guard cells, but in the root, in diagram Y, there's no cuticle, but there will be unicellular root hairs near the root tip. Identify tissues 2 and 3. In fact, what I'm going to do here now, is I'm not going, wait a minute, so I'm not going up and down all the time. So let's do a split. Okay. All right, there we go. So identify tissues 2 and 3. I want this to go away, and it's not going away. There we go. Okay, so identify tissues 2 and 3 in micrograph Y. So there's micrograph Y. That's the endodermis, and the inside layer is the pericycle. Endodermis, to channel or direct the water through the cells. Remember the cells with their Casparian strips of suberin has to go through these cells to the xylem in the middle. And the pericycle, or those cells, that ring of tissue, that cylinder of tissue, is meristematic and produces branch rings. Number 9, which number tissue in micrograph Y is the same as tissue D? Well, D is phloem, and phloem in the root is found in between the arms, the tissue, number 6. The cells labeled 4 and 5 in micrograph Y. So 4 is xylem, looks like little ones, tracheates, 5 is vessels. Okay, name these two. So xylem tracheate, xylem vessels. I put extra information in here. They are xylem. They're both, you're looking at them in cross-section. You're cutting across the length of these cells. Okay, a certain type of plant tissue is shown. Identified. Well, it's phloem. And then the labels, so J is the sieve tube cell, K is the nucleus of the companion cell, L is the companion cell, M is that sieve plate between adjacent, not adjacent, but sieve tube cells placed end to end. Write down the letter or number in micrographs X and Y where this tissue, so phloem, is located. So it's going to be tissue D in the stem and tissue 6 in the root. Okay. Right, then we've got this question that I quickly made up, a little bit different. So this time I said, pupil placed a prepared slide on the stage of a light microscope. After focusing, he saw the following image in the field of view. This would actually be a nice little exam question. Okay, see that up. All right, identify the plant organ. It's the transverse section of a leaf, or just leaf. Is the image the correct way around? No. But we don't give a mark for saying why. All right, so let me just see here. So how do you know this? Using visible evidence. Well, first of all, the palisade cell should be near the top of the leaf below the upper epidermis. I could give 2 marks for that. Or the spongy cells would be above the lower epidermis. Okay, here it's showing the other way around. So I think I've only got 3. Oh, it's out of 3, but I've said it could be out of 4. Yeah, if I even asked you to tabulate it, yes, then it could. Okay, identify tissues labeled B and C. So B are those little ones and C are the long ones. So this is where you could make a mistake. So B is spongy parenchyma and C is palisade, the longer ones, palisade parenchyma. All right, let's move this and go up on the leaf. All right, so B is spongy mesophyll or spongy parenchyma, and C are the palisade parenchyma or palisade mesophyll cells. So I just went and cheated and copied a picture from the internet, but I've got irregular or oval shaped cells with large air spaces. So 1 mark for each of those, 1 mark for naming it, And then C is palisade parenchyma or palisade mesophyll. And then a mark for saying the smaller air spaces, or drawing them with smaller air spaces and pillar-shaped cells closer together. So names of tissues, shape of cells correct, size of air spaces correct. All right, then I said tabulate two differences between tissues A. Okay, this would be quite hard. So I think if you mix them up, we'd probably still just take a 1 mark. The tissue A is actually the lower epidermis and tissue B is the upper. Now, it would be quite nasty if we gave you no marks if you swap them around. So 1 mark for a suitable heading. So table to show the differences between tissue A and D in the transverse section of the leaf. Then a suitable table. Okay, mine's just keywords, but no writing outside the table. Neat ruler drawn table. Okay, so lower, if you said lower leaf epidermis, then there would be more guard cells and a thinner waxy cuticle. Tissue D, the upper leaf epidermis, fewer or no guard cells and a thicker waxy cuticle because that's where the sun shines more. Number 5, which two tissues would form the mesophyll? Well, it's the same ones, so B and C. And I've named them, although you wouldn't have needed to. I was just thinking of different types of questions that could be asked here. Okay. So then we look at the question number 4. In fact, let's move this up. And let's move that, whoops, move that down, and let's go to the questions. Right, so identify tissues X and Y. Well, X is the xylem and Y, we didn't do a lot of this, so phloem or bark, whatever. And what do? So xylem transports water and salts upwards through the tree, okay, whereas phloem transports the sugars or the sucrose that is made in the leaves down to wherever it's needed, or up or down actually. What are labels A, B, and C? They are annual rings. They are rings of secondary xylem, which is actually woody tissue that form every year to increase the width of the tree. So 3 marks there, okay? How old was this tree? Well, I'm not going to count them now, but I counted all these little clear areas and I got 18 years old, okay? Then, which label indicates the time when a drought could have occurred? This one, A, little a, because the annual ring is narrower, so there was less xylem produced because of the water source shortage. And that's the end of that worksheet. So it's quite a comprehensive worksheet, just getting you to go through everything so that it makes a little bit more sense.